He's been very, very productive, as I think you can probably tell.
Yeah.
We have a very interesting group of animals we're working on now. I'm not going to tell you, I'm not going to say anything about it in this talk because it's still very preliminary, but we have managed to engineer a total body knockout of MYC. And the mice have some very, very interesting phenotypes. We're trying to follow them through their entire lifespan. So it's a long-term project, but they basically show very, very profound signs of premature aging and a lot of the associated phenotypes that you might expect with that. Sorry about that. So as I mentioned, it's quite rare, with only about 1 to 200 cases per year occurring in the United States. But the incidence is increasing, and that's mostly because of the improved survival of very low birth weight infants who as a group have a significantly higher incidence of hepatoblastoma. And nearly all the cases are diagnosed before the age of 5, and more than half of these before the age of 18 months. And although survival is quite good, it's very highly dependent on a number of factors such as age, stage, and histologic subtype. And in terms of histologic subtype, actually, HB is classified into 7 distinct groups. And in many cases, 2 or more of these subgroups can coexist within the same tumor at the same time. And I think you can see by these pictures that the presentation is hardly subtle. And for this reason, it's usually diagnosed very, very rapidly. So this is a summary from a Nature article from a couple of years ago that compared the exon mutational frequency of some of the most common forms of adult and pediatric cancers. And what you can see is that pediatric cancers as a group have many, many fewer mutations than do adult cancers. And additionally, hepatoblastoma, which is way over here on the extreme left, has the lowest mutational burden of all. Now, that's not to say that HBs are completely devoid of mutations. In fact, about 80% have somatically acquired mutations in the beta-catenin transcription factor, and 60 to 70% of these have poorly defined dysregulation of the Hippo pathway and its terminal effector protein, which is known as yes-associated protein, or YAP, which, like beta-catenin, is also a transcription factor. And the ultimate consequence of this is that both beta-catenin and YAP are expressed at high constitutive levels and in the nuclei of about half of all hepatoblastomas. In fact, very aggressive tumors that closely resemble hepatoblastoma can be generated in mice by co-expressing mutant forms of beta-catenin and YAP. So how do mutations in beta-catenin activate its oncogenic potential? Well, as shown in this cartoon here, beta-catenin is usually found in the cytoplasm as part of the so-called APC, or destruction complex. And in this setting, beta-catenin is normally phosphorylated by the GSK3 and CK1 serine threonine kinases. And then it's released from the APC complex, and it's rapidly degraded by the proteasome, so that very low levels of beta-catenin are normally found in most normal cells. But then in response to normal growth factor signaling pathway by members of the Wnt family, APC gets recruited to the inner cytoplasmic membrane, where it interacts with the frizzled receptor, and it releases unphosphorylated beta-catenin. And then, of course, this is stabilized. It enters the nucleus, and it interacts with the TCF family of transcription factors, usually most prominently with TCF4. And then this activates many genes that are involved in proliferation and cell cycle progression, such as MYC and cyclin D1. And a somewhat similar mechanism regulates the Hippo pathway, which controls organ and cell size. And this so-called core kinase complex restrains YAP in the cytoplasm, but when it's activated, it releases YAP, which is stabilized. It then also travels to the nucleus, and it activates genes that are involved in growth and survival. And some of these are also beta-catenin targets, and vice versa. So there's a considerable amount of crosstalk between these two pathways. And I should mention that very, very, very rarely hepatoblastomas are found in which there is dysregulation of wild-type beta-catenin without any mutations. But when this occurs, it's usually due to somatically acquired mutations in the APC complex, either APC itself or axin. And the consequences of this are the same in that the same as when you have a beta-catenin mutant, in that the beta-catenin protein is unable to maintain a stable association with this complex, so it moves into the nucleus and constitutively activates its target genes. So oncogenic mutations in the beta-catenin Wnt pathway don't always involve beta-catenin. And in fact, anything that compromises the integrity of this complex is really sufficient to drive tumorigenesis. And there's actually an interesting family cancer syndrome. It's called familial adenomatous polyposis, or FAP, that some of you might be familiar with. And these people have germline mutations in APC. And as young adults, they develop countless colonic polyps, many of which become malignant, and usually by the age of 40 or 45, and this necessitates preemptive total colectomy in order to prevent this from happening. But what's less well appreciated is that these individuals that have FAP have a 5,000-fold higher incidence of hepatoblastoma as young children. And then finally, sporadic garden variety colorectal cancer very frequently involves the somatic inactivation of APC and the activation of beta-catenin. And in CRCs that do not have evidence of APC mutation, they will often carry mutated forms of beta-catenin. So this Wnt beta-catenin pathway actually lies at the center of and links together seemingly unrelated, inherited and sporadic forms of both adult and pediatric cancer.
Okay.
Now, one unusual thing about beta-catenin is an oncogene or oncoprotein is its tremendous mutational heterogeneity. And this just shows a sample of how this can range from anywhere from missense mutations to large in-frame deletions, sometimes as big as 90 amino acids. And the 90 amino acid deletion is the one that we standardly use in our mouse studies to induce hepatoblastomas. And I'll be referring to that throughout the talk as delta 90. So if you hear me talk about delta 90, it refers to the mutated form of beta-catenin that's very highly tumorigenic. And this is really a very different mutational spectrum that we see with other oncogenes. So KRAS mutations, for example, tend almost exclusively to involve codon 12. Although once in a while you'll see mutations in codons 13, maybe 59 or 61. BRAF mutations that are very common in melanomas and thyroid cancers almost always involve codon 600. And in fact, even more restricted is that almost all these mutations involve V600 to E point mutation. So this background information raises a few questions that I think are really key to understanding the mechanisms of hepatoblastoma pathogenesis and behavior. So first of all, why do some patients have localized disease and others have metastatic disease? This is important because obviously advanced disease is associated with lower survival, so we'd like to be able to understand what it is that causes this. Secondly, why do some patients have recurrent disease and others don't? And obviously, it would be nice to be able to identify high-risk individuals right at the time of diagnosis in order to modify therapies right from the beginning, rather than waiting for recurrence to happen. And then finally, what accounts for these 7 different hepatoblastoma subtypes if there are so few mutations? And then a related question, of course, is whether or not beta mutations that have been previously described only in non-hepatoblastoma tumors, such as colorectal cancer or lung cancer, glioblastomas, are they capable of generating hepatoblastomas if they are expressed in the liver? In other words, are certain mutants favored over others in tissue-specific ways, or do they tend to be more agnostic? So we proposed and tested the following 2 major hypotheses. First, we asked whether each beta mutation imparts its own distinct set of tumor properties that could account for the heterogeneity that we observe in these tumors. And then second, the second hypothesis is that all beta mutations are potentially oncogenic regardless of the source. And we tested this by introducing sleeping beauty vectors that encode mutant form of YAP, that Hippo effector, the Hippo pathway effector. And we introduced this along with 14 different patient-derived beta-catenin mutants into the livers of mice using hydrodynamic tail vein injection. And the beta-catenin mutants included the delta 90 one I already mentioned, as well as wild-type beta-catenin plus other point mutations or deletion mutations. And we deliberately selected a spectrum of these patient-derived beta-catenin mutations, including those that had been previously identified only in hepatoblastomas. Those had been identified in other types of tumors, but not HBs, and those that had been identified in both HBs and non-HBs. And what we showed is that each mutant could be classified according to whether the tumors it generated grew rapidly, which we called group 1 tumors, whether they grew at an intermediate rate, which we called group 2 tumors, or at a slower rate, which we called group 3 tumors. And I think what's most obvious about this is that it didn't really matter what the mutant was that we introduced. Every one of them produced some type of hepatoblastoma or hepatoblastoma-like tumor, just growing at different rates. And then the other thing that we found was that each mutation was always associated with its own signature histologic subtype. So for example, you'll notice down here that the delta 90 hepatoblastoma deletion variant always generated what are called crowded fetal histologic subtypes of hepatoblastoma, which is a very common form of childhood disease that we see in patients. And in contrast to that, this S33A point mutation also had a crowded fetal morphology, but it was much more heterogeneous. And then it contained what are called macrotrabecular and blastemal type elements. And then going back down here to the bottom, overexpressing wild type beta-catenin also generated tumors that were mostly crowded fetal HB, but also had a fairly substantial element that resembled hepatocellular carcinoma. And again, based on what I told you before about familial adenomatous polyposis, it's not too surprising that if you force the expression of wild type beta-catenin, then you can generate a tumor. And then each set of these tumors was associated with very distinct transcriptional profiles. So here in panel A, for example, each column represents the mean gene expression differences for 5 representative tumors that were expressing one of 9 different beta-catenin mutants. And what you could see is that the tumors differed from control livers that are shown over here on the left by anywhere from about 4,200 to about 6,800 genes. And then in panel B down here, this shows that there were even more differences were seen if we compared the hepatoblastoma tumors to hepatocellular carcinomas that arise in mice if you overexpress the MYC oncoprotein. And in this case, there were as many as 10,000 differences just between these 2 tumor types. And then up here in panel C, this shows that even when comparing one cohort of tumors to another, so this is just an intra-tumor type comparison or an intramutational type of comparison, the variation among the different beta-catenin driven tumors varied from as few as 1 transcript to as many as 1,100 differences. And then finally down here in panel D, we looked at a group of known beta-catenin direct target genes, and we showed substantial differences not only in the number of the targets that were regulated in response to each beta-catenin mutant, but in some cases, the direction of this regulation. So without going through the many individual studies we did, all of which have been documented in this JBC article up here, what we can say is that the tumor features appear to be driven by the identity of each beta-catenin mutation. And the reason for this is because each mutation determines the absolute level of protein expression, the percent of the beta-catenin protein that's localized to the nucleus, the transcriptional activation potential of the mutant, and the gene expression profiles, as I just showed you. So I think it's fair to say that each beta-catenin mutant that we examined always generated tumors with predictable and unique but overlapping properties, since all these features are ultimately derived from the different gene expression profiles. And so with these experiments, we were able to answer some of the original questions that we had posed, and that had really puzzled us for quite a while, as it had for the rest of the research community. So first of all, why do some patients have stage 1 disease and others have stage 4 disease? And the answer is that, well, presumably because different beta-catenin mutations generate tumors that have very widely different growth rates. What accounts for the 7 different histologic subtypes? Well, we know that each mutant reproducibly generates tumors that have very distinct, reproducible histologic subtypes. How do different beta-catenin mutants affect biochemical, metabolic, and somebody?
कौन-कौन सा? एक नेमाटा है, हाँ, एक इकंडा का, एक धर्मोगी। हाँ, हाँ। अब ये देखना पड़ेगा वो दोनों।
Can you turn off your microphone, please?
कॉपर और स्टील था, ऐसा कुछ था। दोनों चीज हो गई है। हाँ, हाँ। Hello?
Can you turn off your microphone?
Okay.
Turn off your microphone, please. Thank you. How do different beta-catenin mutants affect biochemical, metabolic, and transcriptomic profiles? And the answer is that each mutant imparts distinct but overlapping behaviors. And then can non-hepatoblastoma-associated beta-catenin mutants cause hepatoblastoma? These are mutants that have never been described in hepatoblastoma, but they do arise in other tumors. And the answer is yes, absolutely. And regardless of the source, all beta-catenin mutations and wild-type beta-catenin, for that matter, are oncogenic, at least in the liver. So how else might beta-catenin mutants be affecting the properties of the hepatoblastoma? Well, I just mentioned the 4 major ways that are beta-catenin intrinsic. But what about the possibility of other hits? And you remember that hepatoblastoma is mutationally very poorly endowed. It's the least mutationally altered of all cancers, let alone pediatric cancers. But other hits were still a distinct possibility, and one that we became increasingly more and more interested in. And we were really mainly interested in this because, as I mentioned earlier, but it bears repeating, only about 50% of hepatoblastomas concurrently dysregulate both beta-catenin and YAP. And we know that, at least in mice, neither factor alone is oncogenic. They must cooperate. So it seemed reasonable to postulate that other factors might collaborate with either beta-catenin or YAP and maybe further contribute to the biological and histologic and the metabolic and the molecular diversity that we had already identified. And in the best of both worlds, one might expect that there would be one factor that could cooperate with either beta-catenin or YAP to promote tumorigenesis, rather than separate factors that cooperated with each. So we focused on a protein known as NFE2L2 or NRF2, mostly because after beta-catenin in YAP, its dysregulation in hepatoblastoma and other cancers is actually fairly common. However, it had really not been considered as a causative agent in any of these cancers. And really, it had been studied more in the context of its involvement in mediating antioxidant and detoxification responses. And in terms of its mechanism, like beta-catenin and YAP, it's a transcriptional cofactor, and its regulation is really kind of similar to the other 2 factors in the sense that in normal cells, it resides in the cytoplasm in association with this partner protein, KEAP, or KEAP1. And this interaction occurs via 2 short motifs within NFE2L2, known as the ETGE domain and the DLG domain. And this interaction targets the protein for proteasomal degradation and ensures low-level basal expression. But then in response to electrophilic or oxidative stress, what happens is that cysteine residues in KEAP1 are oxidized, and this causes a conformational change and a release and stabilization of NFE2L2, which then allows it to enter the nucleus. And then once it's in the nucleus, NFE2L2 interacts with these small MAF proteins, which are helix loop helix transcription factors, and it binds to and activates several hundred genes that contain these so-called antioxidant response elements. And the products of these genes are then mainly used to detoxify xenobiotics and to suppress reactive oxygen species. So what we found intriguing is that about 2 to 3% of hepatoblastomas and many, many other cancers contain recurrent missense mutations in NFE2L2. And all of these mutations are located within or immediately adjacent to the ETG, ETGE or DLG domains. And just as intriguing is that as many as 50% of HBs amplify the unmutated NFE2L2 gene. And this is just one study that we did here at Children's Hospital of Pittsburgh using archived tumor samples. And what we showed here on the left is a study in which we quantified NFE2L2 gene copy number from 22 hepatoblastomas. And we showed that 4 of them contained 2 to 6-fold gene amplifications. And then over here on the right is data that we generated from information that was publicly available in the Cancer Genome Atlas. And we were able to show that as many as 5 to 15% of certain adult type tumors also amplified the NFE2L2 gene. So how might NFE2L2 work to either enhance or inhibit tumor growth, both of which, I should mention, have been proposed within the context of modulating reactive oxygen species? And the two models really depend on when, during the course of tumorigenesis, NFE2L2 is dysregulated. So for example, oncogenes such as MYC and RAS are, of course, very well known to generate reactive oxygen species, and then this can be used to drive the additional oncogenic hits that are necessary to fully transform a cell, or at least allow for the evolution of more aggressive or adaptive behaviors. So during these early stages, it's beneficial from the tumor's perspective, at least, not to activate NFE2L2. And that's because that allows the tumor to express high levels of ROS, which drive these additional mutations and tumor evolution. But then later, after most of these important ROS generating mutations have accumulated, the dysregulation of NFE2L2 and the suppression of ROS can now help the tumor by allowing it to increase the expression of those early drivers, which normally would generate such high levels of ROS that the tumor would die. But now that the tumor is able to suppress them due to the activation of NFE2L2, they can be expressed at very, very high levels without mediating any damage and toxicity to the cell, and they can further drive tumor behavior in a very aggressive manner. So in this capacity, NFE2L2 has been proposed not to really function as much as an oncogene as it does as what I call a tumor facilitator. And then in the second scenario, NFE2L2 serves what I call a tumor inhibitor to distinguish it from a tumor suppressor. And again, just as in the first model, it really impacts tumor growth indirectly. And it's this that distinguishes it from the true tumor suppressor. And then according to this model, what happens is that early NFE2L2 activation here suppresses ROS right from the beginning. And that, of course, will tend to mitigate the emergence of mutated drivers. And if those driver mutations have already been generated, then NFE2L2 minimizes the subsequent rate of ROS-dependent oncogenic dependent evolution and the natural selection of the most aggressive tumor subclones. So if you do develop a tumor, then it's likely that it's going to be much less aggressive than the tumors that derive from this first model. So we asked then what would happen if we simultaneously co-expressed the delta 90 beta-catenin and YAP together with either wild type NFE2 or one of two patient-derived missense mutations, one that I'll call L30P and R34P. And both these mutants, the first mutant, the L30P, involves the leucine within the DLG domain. That's the motif, remember, that interacts with KEAP1. And the R34P mutant is just a couple of residues away from that. And what you can see from this picture is that both mutants, but not the wild type NFE2L2, very significantly accelerated the growth of these delta 90 plus YAP tumors that were already very rapidly growing. This was one of the most rapidly growing subsets of tumors that we had ever seen. So at least in this particular context, these findings seem to rule out the notion that NFE2L2 is acting as a tumor inhibitor. In fact, it seems quite the opposite, given how rapidly these both of these mutants accelerate the growth of these tumors. And what was unexpected, though, is another feature of these tumors, which is shown here, which is that both L30P and R34P, but again, not wild type NFE2L2, in addition to promoting rapid tumor growth, they produced a very striking tumor phenotype, which included the presence of these innumerable and in some cases quite large hepatic cysts or intratumor cysts, which were associated with these very large areas shown here by the arrows of very extensive pericystic necrosis. These cysts were bloodless, and they were not lined with endothelium, but rather they were lined with tumor epithelium that stained very intensely for nuclearly localized beta-catenin. And this type of cyst actually has been previously described in very, very rare cases of hepatoblastoma, but not nearly to the extent that we found it to be present in these mouse tumors. So these findings were initially more compatible with the notion that NFE2L2 mutants were serving as tumor facilitators. But really, this notion was also proved wrong by these experiments, in which what we did here was that rather than co-expressing all three culprits together, we expressed them either individually or in all possible pairwise combinations. And what we found was that none of the individual factors shown up here, either delta 90 alone, YAP alone, L30P alone, R34P alone, none of these individually was oncogenic. And this, of course, is what we had expected and seen many, many times before. But on the other hand, every pairwise combination, no matter what the combination, every pairwise combination was oncogenic and was associated with distinct tumor histologies. So what this showed was that unlike the tumor inhibitor and tumor facilitator models I showed you a couple of minutes ago, and which may well be true for the wild type NFE2L2, the L30P and R34P mutants were acting in very different ways. And in fact, they were really acting like classical oncoproteins, just like the mutant forms of delta 90 and YAP do. And now we did a lot of metabolic and biochemical profiling of these tumors to characterize them. And I'm only going to show you only one set of results, but I think it makes a general point. And in this study, what we did here was to look at OXPHOS in partially purified mitochondria from tumors that were generated by all those different possible combinations of delta 90, YAP, and L30P and R34P. And we measured the oxygen consumption rates in response to various complex 1 and complex 2 substrates, as well as to glutamine and the fatty acid palmitate. And what we had previously seen in tumors that were generated only by delta 90 and YAP is that oxygen consumption rates tend to be much lower in all cases relative to control livers that are shown over here on the extreme left. This was almost always the case. And this lower oxygen consumption was seen in response both to complex 1 substrates such as pyruvate and malate and to complex 2 substrates such as succinate. And you can see that glutamate and palmitate responses were also significantly downregulated in most of these tumors as well. And these reductions were in keeping with the idea that rapidly growing tumors upregulate the Warburg effect and downregulate OXPHOS. But unexpectedly, what we found was that the combination of YAP and either L30P or R34P generated an increase in OXPHOS relative to the liver, as indicated here in these blue boxes. So not only do beta-catenin and YAP mutants generate different responses based on the nature of the beta-catenin mutations, but each factor also provides very different metabolic profiles in response to the dysregulation of NFE2L2. And the gene expression profiles of the delta 90 plus YAP tumors were also quite distinct, depending on whether they expressed wild type or mutant NFE2L2. So this is a heat map for the total gene expression differences among classical HBs that are expressing delta 90 and YAP, and that's shown over here just in this left-hand most panel. So just these two oncoproteins, delta 90 beta-catenin and YAP. And then the middle column here shows the same tumors, but now they are co-expressing wild type NFE2L2. And then the right column shows the same tumors again, now expressing, instead of wild type NFE2L2, they are expressing either L30P and R34P. And I should mention that the results of the L30P and R34P were combined because they were absolutely identical. So this column contains 10 samples, the average of 10 samples, and the others are all the average of 5 to 6 samples. And a couple of things are apparent. So first of all, despite having no discernible effect on any of the tumor properties that we examined, wild type, somebody has their microphone on. Could you please turn it off? Wild type NFE2L2 was not entirely without effect, and it was actually was associated with the dysregulation of over 800 transcripts. And in contrast, the L30P and the R34P expressing tumors not only deregulated twice as many transcripts as did the wild type, but in some cases they did so in opposite directions, as shown here by these black boxes. And depending on which data set we use, somewhere between 5 and 22% of all the differentially expressed transcripts were the products of previously confirmed direct NFE2L2 targets based on ChIP-seq results. So at the end of the day, we've shown that every possible combination of beta-catenin, YAP, and NFE2L2 mutants is oncogenic. But we're still left with the nagging and, in my mind, most important question, which is, with more than 5,000 gene expression differences between livers and tumors, which of these are actually really mediating transformation? And despite knowing that these 3 different transcription factors are capable of driving transformation in very distinct ways with any pairwise combination, it really tells us very little about the mechanisms by which they do this. So given how many tumors we had examined here and in the past that were driven by all sorts of different beta-catenin mutants and YAP and L30P and R34P, we decided to take advantage of their very distinct gene expression profiles by doing what I call triangulating their transcriptomic profiles to see if we could pinpoint a common set of targets that were deregulated in all cases, under all circumstances, and regardless of any other features and behaviors of the tumors. Now, to do this, we used a very stringent cutoff criteria, and we used 3 different methods of analyzing the gene expression differences. We were very conservative in our analysis in that we proceeded with only those genes that were identified as being differentially expressed by all 3 methods. And this allowed us then to classify genes that were regulated by only a single factor, by 2 factors, or by all 3 factors. And we called this latter group BYN targets, beta-catenin, YAP, NFE2L2, because they were regulated by all 3 sets of factors. And you could see that there were only 41 of these common factors, 30 of which were upregulated and 11 of which were downregulated. So then, based on our previous RNA-seq results of tumor sets that were generated by 9 different beta-catenin mutations, we asked how many of these BYN transcripts were always regulated in the same direction, that is, always up or always down regulated in tumors relative to livers, regardless of the underlying drivers. And then with these criteria, we managed to identify 29 of these 41 transcripts as always maintaining the same unidirectional expression. And then we asked how many of these transcripts, of these 29, were deregulated in tumors that were generated by the same beta-catenin mutant that is delta 90, but now generated in hepatocytes that lacked two key transcription factors that were determining how rapidly the tumors grow. And those were MYC and a member of the MYC network known as ChREBP. And the reason, as I said, the reason that we did this was because we had shown previously that if you generate tumors in hepatocytes that lack those two factors, that the tumors grow very, very slowly. You still get tumors, but they just grow very, very slowly. So doing this allowed us to eliminate an additional 7 factors that left us now with 22. So let me just reiterate, using very extremely stringent criteria, we identified a core set of 22 transcripts that was always deregulated among multiple tumors and always in the same direction, irrespective of the identities of the oncoprotein drivers and regardless of tumor growth rates. We were very, very particular about this last criterion, since we were more interested in identifying genes that were responsible for tumor initiation rather than tumor growth rate. So what do these genes look like? Well, of those 22 BYN transcripts, 16 were linked to at least one, and in some cases as many as 7 to 9 of the 10 known classical cancer hallmarks that were enunciated a number of years ago by Doug Hanahan and Bob Weinberg. And to put this into context, 5 very well-established oncogenes or tumor suppressors shown over here on the left, and I'm sure which all of you are familiar with. These were associated with somewhere between 5 and 8 of these hallmarks. So our BYN transcript seemed pretty competitive when put up against this very impressive time-tested group. And then another interesting and gratifying feature of BYN transcripts that we had identified built upon 2 previous reports. One report showed that hepatoblastomas with a particularly poor prognoses and outcomes could be identified a priori based on the expression of only 16 transcripts. And then the second paper elaborated on that first report by showing similar findings that relied on the identification of only 4 transcripts, none of which were related to the previous 10. So what we found was that 10 of these BYN transcripts, also having no overlap whatsoever with the just mentioned 10 and 14 transcripts, also perfectly identify these bad actors shown over here on the right and labeled C2A and C2B. These are all the patients with unfavorable prognosis. And then finally, we show that every one of our 22 BYN transcripts was the most highly dysregulated when tumors co-expressed all 3 oncoproteins, that is delta 90, YAP, L30P, or R34P. And that's labeled over here in red, and you can see their degree of up or down regulation. The BYN transcripts were the next most highly dysregulated in delta 90 plus YAP tumors, shown over here by column number 2, and they were the least dysregulated, but nevertheless still very significantly so, in tumors that were generated either by the combination of YAP and NFE2L2, shown over here, column number 3, or the delta 90 plus NFE2L2 mutants, column number 4. We also showed that 17 of these 22 transcripts correlated with survival in a large number of adult cancers, shown over here on the right, based on the expression and survival data that was taken from TCGA. And in a couple of these cases, this was true for as many as 6 or 7 different tumor types for any given gene. And then the other thing I would mention is that the transcripts shown here that were tagged with these red asterisks were also significantly upregulated in delta 90 plus YAP tumors that were co-expressed with wild type NFE2L2. And since wild type NFE2L2 is not oncogenic, or it doesn't contribute anything, I should say, to the tumors that are formed by delta 90 and YAP, this suggests that these genes may be less likely to be the true drivers of transformation than the non-star genes are. So perhaps we should maybe be focusing our attention, at least initially, on the remaining 8 non-star transcripts, which would further narrow the list. So then we went to the ENCODE database to determine whether any of these BYN target genes were direct targets for the 3 transcription factors. And again, depending on the source of the ChIP-seq data, we showed that the 5 kb of proximal promoter for the vast majority of the genes that we looked at contained bona fide TCF binding sites for beta-catenin. They contained TEAD sites for YAP, but only 2 promoters contained ARE sites for NFE2L2. So the gene promoter that's shown here by this red arrow is a real outlier in the sense that it contains many more documented binding factors for each, or binding sites for each factor than does any of the other promoters. And it's one of only 2 genes that contain any NFE2L2 binding sites at all. So what is this gene? Well, it's a gene called Serpin E1. And it's the second most highly upregulated gene in hepatoblastomas. It's upregulated by 44-fold relative to the liver. And although this figure that I showed you a couple of minutes ago indicates that Serpin E1 is involved in at least 5 different cancer hallmarks, the protein itself was originally studied in the context of its role in lysing blood clots. So for this reason, it was previously named plasminogen activator inhibitor 1, or PAI1, to indicate that historical association. And since its canonical role in fibrinolysis was described, it subsequently has been shown to play important roles in tumor growth and metastasis and angiogenesis and matrix remodeling. And we were particularly intrigued by these non-canonical functions because actually high levels of circulating Serpin E1 are associated in young women with polycystic ovary syndrome, or PCOS. And in fact, if you express Serpin E1 in the ovaries of mice, they will actually generate cysts that very closely resemble the cysts that you see arising in the ovaries of women that have polycystic ovary syndrome. So to get at that question, we first looked a little bit more closely at these tumors to see if, in addition to high levels of Serpin E1 transcripts, we could also document high levels of the protein. And indeed, the answer is yes. As you can see here on the extreme left-hand side, both control livers and most HBs that we looked at contained very low to undetectable levels of Serpin E1. The exceptions were tumors that expressed either L30P or L34P, along with delta 90 and YAP. So the combination of delta 90, YAP, and NFE2L2 mutants was needed both for the highest levels of Serpin E1 to be expressed, as well as for the generation of cysts and necrosis. And on the right are Western blots showing that although very little Serpin E1 can be detected in the serum of tumor-bearing mice, the cyst fluid contains very high levels of Serpin E1. And I should mention that the patterns of Ponzo red staining that were performed on serum and cyst fluids look very similar. And what this indicates then is that the cyst fluid is made up of a transudate from the serum, and that's very typical of most cysts. So it appears that the surrounding tumor cells synthesize Serpin E1 at very high levels. They secrete it directly into this transudate, but very little of it gets out and enters the circulation. And then finally, we asked whether the enforced expression of Serpin E1 could substitute for L30P or R34P when it was co-expressed in tumors with delta 90 and YAP being co-expressed as well. Now, that is a very tall order because remember that NFE2L2 mutants alter the expression of 21 other BYN transcripts. So it would be unlikely that any one of them might recapitulate all these effects, but we at least reasoned that maybe we might see some additional phenotypes, and it seemed like a good place to start. So we generated hepatoblastomas with delta 90 and YAP with a Sleeping Beauty vector that also expressed epitope-tagged Serpin E1, and then we measured the Serpin E1 and lysates from each tissue. And here on the top in the first two lanes is just a repeat confirmation that control liver expresses very little, if any, Serpin E1. The next two lanes show that the control delta 90 and YAP hepatoblastomas expressed maybe a little bit more, but still very, very little. And the next two lanes show that Serpin E1 protein levels are again higher in tumors, driven by delta 90 YAP and L30P or R34P, as I showed before. And then the last two lanes show that delta 90 YAP and Serpin E1 expressing tumors express both the endogenous and the exogenous epitope tag Serpin E1, as indicated here by its slower mobility in the gel. And although the tumors that co-express Serpin E1 did not grow any faster or develop any cysts, they did contain these very large areas of necrosis that were a very prominent feature of the original BYN tumors that I showed you before. And H&E staining confirmed the presence of these very large areas here of necrosis indicated by the arrows that were scattered throughout the tumor parenchyma, although we really didn't see any cysts associated with them as we had sort of expected we might. So, but what I think these experiments tell us is that while enforcing Serpin E1 overexpression doesn't fully recapitulate all the tumor phenotypes that you see when all three transcription factors are co-expressed together, it does at least alter the tumor in a way that begins to resemble this state. And as was probably apparent from the beginning, more than one member of this group of BYN genes very likely needs to be coordinately dysregulated in order to recapitulate all the morphologic features of the tumors that are generated by beta-catenin, YAP, and NFE2L2 mutants. In fact, multiple BYN genes probably require dysregulation simultaneously. So we would predict that as we dysregulate more and more of these BYN transcripts in the appropriate way, more of the tumor phenotypes might emerge. And if you take the most reductionist approach possible, we would predict that if these 22 genes were dysregulated in precisely the manner shown here, then we should theoretically be able to generate hepatoblastomas in a way that circumvented the need for having to express beta-catenin, YAP, and NFE2L2. And that's sort of what we're working on at this point. So the final conclusions then, just to summarize everything, are that NFE2L2 is dysregulated in as many as half of HBs and up to 15% of adult cancers. Mutant forms of NFE2L2 are directly oncogenic and don't just modulate tumor growth. And the oncogenicity of NFE2 requires both its mutation and the co-expression of either beta-catenin or YAP mutants. And then in combination, mutant NFE2L2, beta-catenin, and YAP generate tumors with unique properties that have to do with growth rates, cyst formation, necrosis, altered metabolism, distinct transcriptional profiles. Beta-catenin, YAP, and NFE2L2 mutants share a common set of these so-called BYN target genes that are highly deregulated in hepatoblastomas and many other adult cancers. And 82%, 18 of 22 of BYN transcripts are predictive of survival in hepatoblastoma or other adult cancers. And the ectopic expression of Serpin E1 recapitulates the necrosis that is seen in tumors generated by the combination of NFE2L2 mutants, beta-catenin mutants, and YAP mutants. And finally, the full spectrum of properties of the other beta-catenin, of the other BYN transcripts, both alone and in combination, remains to be determined. And as I mentioned, this is what we're currently working on. So with that, I'm just going to close. I want to recognize the people in the group very briefly who did all the work. Most notably, Huabo, who Lokendra knows quite well, has been an absolute workhorse and has been the person who's been most responsible for driving this and overseeing the work of a lot of the other people. Wei Shi is a student who came to my lab for a couple of years from Tsinghua University. She and Jenny Mayfield, a medical graduate from the University of Mainz in Germany, both together worked on all of the experiments showing that various types of beta-catenin mutations could impart distinct behaviors to tumors. They were ably assisted by my laboratory manager, Ji Lu and Sucheta over here. Had a number of people that worked very hard on the bioinformatics analysis of our tumors. That included Jordan, and then 3 absolutely outstanding medical students. James, who is currently a hematology oncology fellow at my alma mater, the University of Chicago, and then Ying and Brady, who are have just begun their residency training. Ying at NYU in New York, and Brady here, fortunately, at Children's Hospital of Pittsburgh. Joanna Gorka was an undergraduate in the laboratory who worked with us for 4 years until she took a Fulbright fellowship and left to spend a year working in Poland. So with that, I thank you for your attention.